think we'll go ahead and get started here. So we're going to today conclude a full hour on ligand target interactions. And what we're going to go through today is something that's very important in pharmacology, which is selectivity. How selective is a drug for a particular receptor? And you know, the situation is there are just thousands of receptors in the human body. And so the best drug will have a high affinity for one and only one receptor. Imagine a drug that just activates all your receptors in your entire body. What kind of a day would you have after you took such a drug that just tweaked out and turned on all the receptors in your body? You'd probably be dead in like 3 minutes, you know? So, or a receptor that activates, or a drug that activates all the receptors in your brain, all thousands of them. You would have a very bad day. So we want drugs that have high selectivity, but the problem with biology is that there are many receptors, there are families of receptors in the body in which receptor A is very similar to receptor B, but not quite identical, which is quite similar to C and D. And so we're going to use a real-world example today in the second half of the lecture from neurobiology and neurotransmitters. So this is going to be kind of like a brain-focused set of lectures today because there's so many receptors in the brain, but it provides a great opportunity to illustrate what selectivity really is. Okay, so basically I'm going to try to illustrate what drug selectivity is using an actual biological scenario, which is brain chemistry, brain biology. So here we go. So as I just said, we're going to talk a lot about serotonin and its receptors today, and we'll illustrate drug selectivity using this real-world example. So in order to do that, I don't want you to be most people think that like neurobiology is really difficult, and it's true, the brain is complex, but the same principles operate in neurobiology as in any other organ system, so we'll just use that. So most people are indeed familiar with what neurotransmitters are. These are small organic compounds that are made by neurons. Examples are like serotonin, dopamine, norepinephrine, epinephrine. We're going to focus largely on serotonin and dopamine today. But you can see that these are just kind of smallish organic molecules that are ligands, and they're ligands that bind receptors in the brain. So for instance, there are a whole family of serotonin receptors, and they bind serotonin. But each one of these receptors has a different affinity for serotonin. So some might take only a tiny bit of serotonin to become activated, and others might require a whole boatload of serotonin to become activated. So these serotonin receptors come in families, and there are many related receptors. Interestingly enough, actually serotonin is a bioactive amine, but some serotonin receptors also have affinities for dopamine as well, also an amine. So you have receptors that themselves can be activated by either serotonin or dopamine, both, but they might have differing affinities for serotonin as opposed to dopamine. So there's some complexity in the system, right? And of course, serotonin receptors are very important medically because they're tied to anxiety and depression, schizophrenia, obsessive compulsive disorder, PTSD, panic disorder, a lot of really important behavioral health disorders. And so if you look at what these things roughly do, here is an idea, here is just kind of a Venn diagram showing these three major neurotransmitters, serotonin, dopamine, and norepinephrine. And you can see that, for instance, OCD, obsessive compulsive disorder, is very highly regulated by serotonin levels. Dopamine affects alertness and energy. Norepinephrine is involved in attention, motivation, and pleasure rewards. But anxiety is influenced by both serotonin and dopamine. And mood is influenced by all three neurotransmitters, okay? And that's because there's a complexity of receptors in the brain. So not a simple situation. So if you want to develop a drug, you want to find a drug that's going to be very selective. The medical significance of serotonin, I just kind of almost just said that, but serotonin is a neurotransmitter, and the levels of serotonin in your brain are tied to a number of important conditions. The most famous of which is probably depression. Most people know that serotonin, low serotonin levels can lead to depression, also anxiety, but more severe disorders like schizophrenia also tie in with serotonin, OCD as well, panic disorder, post-traumatic stress disorder. So let's get into serotonin itself now and talk a little bit about how serotonin is made. So it turns out now we're just talking about the biosynthetic pathway that leads to the production of serotonin, and then we're going to talk about how serotonin is degraded and destroyed metabolically. So serotonin, also called 5-hydroxytryptamine, so 5-HT is the same as serotonin. It's synthesized in two biochemical steps, and the starting material is simply the amino acid tryptophan, L-tryptophan. So tryptophan is acted on by tryptophan hydroxylase, and it turns it into this L5-hydroxytryptophan, which is then acted upon by another enzyme, a decarboxylase, and that yields 5-HT, or serotonin. So two steps, mediated by two different enzymes. And you can see that there are some cofactors that are involved with the enzymes to make this happen. So, Thanksgiving coming up. Turkeys are loaded with tryptophan, right? So if you eat the entire turkey yourself, you can make enough serotonin to have an effect on you, though it's a challenging thing to eat a whole turkey. So that's the biosynthetic pathway. Now let's look at the degradation pathway, what's downstream from serotonin. So remember, here's serotonin, and it turns out that serotonin can be destroyed by enzymes called monoamine oxidases, MAOs. And interestingly enough, there's an acetylase that can also turn serotonin into melatonin. Maybe that's why the turkey makes you sleepy, right? Because you eat the tryptophan, you make serotonin, and then there's a pathway that leads to melatonin, which makes you very sleepy. So two things can happen to serotonin once it's made, and that's shown here. So here's serotonin again. Let's follow this rightward pathway first. Serotonin can be turned into this entity in acetyl 5-HT, and then that can be turned into melatonin by a couple of steps. That's not our primary interest here. We're interested really in therapeutically. It turns out that this pathway over here is also very important. So it's kind of split into two sides, but remember, the key enzyme that turns serotonin on this slide into 5-hydroxyindole acetaldehyde is this MAOI. So MAOIs turn serotonin into this entity. And that's no longer serotonin, so it's no longer going to activate serotonin receptors. Okay? So, if you have an enzyme, MAO, monoamine oxidases, that turns serotonin into something else that's no longer active, here's a question for you. MAOs are pretty important. They're destroying serotonin, right? What happens if I find a drug that inhibits MAO, an MAO inhibitor? What would happen to the levels of serotonin? I see people pointing up, that's the correct answer. Serotonin is going to accumulate because you've inhibited the thing that destroys it, right? So its levels will accumulate. So MAOIs, monoamine oxidase inhibitors, should increase serotonin levels, right? And guess what? That could have a positive effect on depression, for instance. And indeed, MAOIs were really the first drugs developed that specifically targeted the serotonin pathway for that reason. So that gets us into the pharmacology. How do you target serotonin and its receptors pharmacologically to achieve a beneficial outcome, say to treat depression, for instance? There are actually three main strategies. One of them I just mentioned, inhibition of serotonin degradation by using drugs that inhibit monoamine oxidase activity, so serotonin levels will rise. There is also these serotonin functions in a synapse, and serotonin can be retaken up into neurons by a transporter. So if you inhibit the reuptake of serotonin, that also causes levels of serotonin in synapses to increase. And that is achieved pharmacologically by SSRIs, which are called selective serotonin reuptake inhibitors. There's that word, selective, right? So you wouldn't want a good SSRI is not going to also inhibit dopamine pathways, for instance. So selectivity is important. The third main approach is you can actually, serotonin activates serotonin receptors, so perhaps you can come up with things like agonists or antagonists that work on the serotonin receptors themselves. So three different ways you can approach this from pharmacological perspectives, and all three have led to drugs that are currently in use. Let's talk a little bit more about serotonin reuptake. So here is a diagram of two neurons and a synapse. So synapses are simply a little bit of space between two neurons, and it's where signaling from the upstream neuron takes place to signal a downstream neuron. So we would call, and this is the usual way these diagrams work, this is the presynaptic nerve, and this is the postsynaptic nerve, and the space in between is the synapse. And so this neuron up here can trigger this neuron down here to fire, or signaling from this, and that would be, an activation situation. Sometimes synapses inhibit the neuron downstream to fire, and that's an inhibitory synapse. But it's all mediated by the release of neurotransmitters. Okay, and so we're talking about serotonin. So where does serotonin come from? Well, it certainly comes from that biosynthetic pathway, and that's happening up here in this presynaptic neuron. But once serotonin is made, it's placed into vesicles, and then the vesicles fuse with the presynaptic cell membrane and release their contents into the synapse, okay? And so serotonin is released into the synapse. And then it can bind serotonin receptors in the postsynaptic neuron and tell that neuron to do something, okay? Like transmit a brain signal, right? But serotonin gets recycled. So serotonin can also be taken up by a transporter in the presynaptic neuron, and it's taken out of the synapse and put back into the presynaptic neuron where it can be reused, okay? So we're thinking about inhibiting stuff in a biochemical pathway. Now let's consider inhibition here. Here is the transporter that takes serotonin out of the synapse and puts it back into the neuron where it isn't functional, right? What happens to serotonin levels if we inhibit this reuptake transporter? What do you think? Okay, a lot of people going like this. Anybody's going like this, you're all right, okay? Good feedback. So if you inhibit that, serotonin is going to accumulate in the synapse, right? So if depression is, broadly speaking, a disorder caused by low levels of serotonin in synapses, you give them an SSRI and serotonin levels increase and you can alleviate the disorder. That's how that works. And SSRIs are things like, there are a bunch of them. Okay, so that's some interesting stuff. We talked about monoamine oxidase inhibitors briefly. We talked about SSRIs. But now we want to get into the business of these serotonin receptors themselves. And this is going to get a little bit more complicated. So there isn't just a single serotonin receptor. In reality, there is a family of serotonin receptors. And this is how they're named and described. So 5-HT is serotonin, right? So there's the 5-HT1 receptor, the 5-HT2 receptor, 3, 4, 5, 6, 7. So at least 7 different kind of families of, or subtypes of serotonin receptors. And you can see that some of them are excitatory in synapses and some of them are inhibitory. And these are primarily on the postsynaptic neuron. That's where they're expressed. They happen to be G-protein coupled receptors. That's a certain type of receptor, the most common type. And we'll spend a couple of days working on those later on. They're found in both the central nervous system and the peripheral nervous system. And they can be excitatory and inhibitory. So for instance, an excitatory serotonin receptor like 5-HT2, when it binds serotonin, it'll cause the downstream neuron to transmit a nerve impulse. Whereas an inhibitory one, like 5-HT1, when that one binds serotonin, it causes the downstream neuron to be inhibited. So these are obviously different kinds of neurons downstream, right? Some are excitatory, some are inhibitory. And the other thing that's not shown on this slide, but is very important, is that there are many types of neurons in the brain, and the brain has many specialized regions, and some regions of the brain might express certain serotonin receptors, and other regions of the brain might express a different set of serotonin receptors. So there's an anatomical difference in the distribution of serotonin receptors as well. So there's a lot going on here, right? But it's not too hard to understand. So how have people figured out how serotonin receptors work? Well, they worked with laboratory mice, and serotonin receptors are encoded by single genes. So in mice, people have deleted specific serotonin receptor genes, and then you see what happens to the mouse, right? These are knockout mice. And so this table here shows where all of these different serotonin receptors have been knocked out in mice and what the phenotype is, what the consequence of deleting that receptor is. So for instance, if you delete the serotonin receptor 5-HT1A, now that results in increased anxiety in the mice, right? So if you knock out the receptor and you get increased anxiety, what does the receptor do when it's intact? It reduces anxiety, right? So 5-HT1A prevents anxiety from happening. You knock it out, you get anxiety. Make sense? So the actual function is always opposite of the phenotype that you get when you knock it out. Okay? And here's 5-HT1B. When you knock that one out, you get aggression. When you knock out HT2A, you get anxiety. When you knock out 5-HT2B, that's lethal because that serotonin receptor actually works in the heart. So it's not even in the brain. And so, you know, this previous slide, I showed you all these different families, 5-HT1A2. These are actually families of receptors. And within each one of these families of receptors, there are actually other additional receptors that are all in the same family. Like 5-HT1A is a receptor, 5-HT1B is a second receptor, but it's in the 5-HT1 family, okay? So there are families and subfamilies of these receptors. It's a complex situation. But by knocking them out one at a time in mice, you can figure out what they do. And they control things like anxiety, aggression, heart function, appetite, seizures, pain nociception, which I think is pain experience, behavior, things like exploration, sensitivity to ethanol, regulating body temperature, all sorts of stuff, right? all mediated by a huge milieu of serotonin receptors. So, drugs have been developed that are fairly selective for specific receptors, and here are some examples of them. So, receptors that work on 5-HT1A, remember 5-HT1A, when you knock it out, you get anxiety, right? So let's look at the drugs that are agonists of 5-HT1A. If you knock it out, you get anxiety. So if you had an agonist as a drug that activates that receptor, you're going to decrease anxiety, right? And that's why these drugs work. So for instance, here, these are partial agonists, means they activate it to some degree, but drugs such as buspirone and ipsaparone are drugs that agonize the 5-HT1A receptor. They activate it to some degree. Here's another drug, somatropan, treats migraines. Here are some drugs here, ketanserin, risperidone, work on depression and schizophrenia. And so you can go down this list and see different drugs that work in the serotonergic pathways. And down here at the bottom are selective, these are ones that work on the transporter, so not the receptors. So those are the SSRIs, for instance. Things like fluoxetine, for instance, which is, I believe, Prozac. Okay, so we need to now get into, think about these drugs. They've been discovered by screening through thousands of different drugs and finding the rare molecule that hopefully is pretty specific for one receptor, even though there are many, many other similar receptors present, right? How do you end up dealing with that type? So selectivity is really important. How do you deal with that? Okay, so selectivity is just a really important concept, and it's probably the most important concept for you to learn in today's lecture. Ligands, that can be neurotransmitters themselves or drugs that also bind receptors. They can be selective or non-selective. So selective means one ligand binds one and only one target, and vice versa. Non-selective, one ligand can bind many targets. Or you could say one target, one receptor might bind multiple ligands. Those are both examples of non-selectivity. But selectivity means nothing by itself. It has to be a comparison. Like I could say, is this screen here white? Well, it's white compared to this black table, but it's not particularly white compared to the sun, right? So you have to do a comparison in order for this to be meaningful. And the same has to do with drug selectivity. It's only a term that has any meaning if you're comparing one interaction as compared to another interaction, one affinity compared to another affinity. So here are a bunch of serotonin antagonists, and here are dopamine antagonists. So these are all drugs that would antagonize serotonin receptors. These are drugs that would antagonize dopamine receptors. And you can see that there are several molecules here that are kind of specific for serotonin receptors. There are other drugs over here that are specific for dopamine receptors. And then here are a couple of molecules that inhibit or antagonize both serotonin and dopamine receptors. So these are the ones that have overlapping activity. These are the non-selective ones, right? Okay, so how do we measure selectivity? I'm going to put something on the board quickly. It turns out we use a lot of times this, terms like PKI to describe numerically what these affinities are. So I want to help you out with this a little bit. So you remember KD, we were talking about that last time, right? So KD is a concentration. And so Ki is similar to KD. It's just that this one was determined with one of these competition experiments. So you can actually mathematically, we would say pKi is equal to the negative log of Ki. Okay? So remember KIs, these are concentrations. So we might have, let's just illustrate this. Low affinity, high affinity. Let's compare two drug receptor scenarios. One's low, one's high. Low affinity, the Ki, for instance, Ki might be 10 to the minus third molar. That's 1 millimolar, right? A high affinity one might be Ki equals 10 to the minus ninth millimolar. So all you're doing when you use this equation, pKi equals negative log of Ki, would be the following. So pKi of this one is equal to the negative log of that number. Well, the log is minus 3. Negative means change the sign, so that becomes 3. The pKi of this one, the log, is 10 to the minus ninth. So pKi, the exponent is minus 9. So pKi is 9. So this is high affinity. pKi 9 is high affinity as compared to pKi 3, right? So that's just kind of how that works. So that's just to kind of help you out a little bit here as we go through this slide. So it's the magnitude of difference between the binding of two different ligands to a particular target. Or it could be two different targets with one ligand. And that's kind of probably more important in terms of pharmacology and selectivity. So let's look at this. Here is, let A be a target, B be a ligand, and C be a different ligand. So here's one target that gets together with ligand B, and you have an AB complex. Here's the target, a receptor called, a target called C gets together with this ligand A. Now you have an AC, okay? So in this case, we have one ligand and two different targets called B and C, okay? We have two different ligands and one target. So this would be like comparing two different drugs called B and C and their affinity for a target receptor called A, okay? So here's a second comparison, But anyway, here's the idea. Selectivity, so if the AB complex, target A complex with drug B, we're going to compare that to receptor A complex with target C, or excuse me, ligand C. So B and C are the different drugs, A is the receptor. How do we compare that in terms of selectivity? Well, we could take the Ki of one interaction and compare it to the Ki of another interaction, okay? And so if remember, these are basically, the Ki's themselves are actually exponents, right? Like minus 3 and minus 9. So if you divide these things, say you had, let's say this one is actually the -9, that's -3. When you divide exponents, you actually subtract them from mathematics, right? In this case, though, we have a different set of numbers. pKi of the AB interaction is 8. pKi of the AC interaction is 5. So we've already done the negative log thing. 8 minus 5 is 3, right? So the difference is 3 orders of magnitude, 3 exponents, right? And so that means that A is 1,000 times more selective as compared to the other interaction. You get the idea, right? So we're going to use KIs, for instance, and PKIs, this type of language to describe different affinities. And now we can put a number on selectivity, okay? So let's go through an example. So here is an agonist of serotonin receptors, and it's called ketonserin, and here's its structure, and here's serotonin itself. And so this is a serotonin receptor antagonist that is used to treat hypertension. This drug is an antagonist of the serotonin receptor 5-HT2A, but it also has a little bit of activity against 5-HT2C. And it also kind of, to some degree, binds histamine receptors and alpha adrenergic receptors. So this drug here can bind a lot of different targets, so not terribly selective. But a big question is, how much are those differences? Here's a look at it. So the ligand binding domain is on the surface of the protein. Here we're looking at the receptor. And what we're showing here is actually just kind of the basically the protein structure of a receptor. But what we're seeing here is this drug binding the receptor, and the drug is shown in red. So of course it has some kind of a structure-activity relationship. Okay. So what does this look like chemically? So here is 5-HT2A, the receptor, and that's all the stick figure components of the relevant amino acids in the receptor. And here's serotonin. And here is ketonserin in a similar region of the receptor. And you can see that they both bind, basically, but they probably do so with rather different affinities, slightly different properties. Here's another example, buspirone. Here's serotonin, and here's buspirone. And you can see that, these drugs, this part here looks kind of like serotonin, but not exactly. It's used for general anxiety disorder, and it's a partial agonist of 5-HT1A. Remember, that's the receptor that controls anxiety, right? But it also is a bit of a dopamine agonist. So it's another example of a drug that has only partial selectivity. Sometimes a little bit of crosstalk is good pharmacologically too. You can take one drug that does two things. You don't have to take two drugs so long as it's targeting the right intended things. But here we are now, and I want to just kind of get into this in a little bit more detail. And what we have here are some results of scientific studies that are measuring affinities using pKis. And let's look at what we have here, kind of a complex table. Up here at the top, we have 3 different serotonin receptors called 5-HT2A, 5-HT2B, and 5-HT2C, okay? And in this table, we're then going to look at a number of different drugs. So spipirone, for instance, this is a drug that can bind serotonin receptors. And we have a value of 8.8 for 2A, 5.5 for 2B, and 5.9 for 2C. What does that mean? Well, since this is a pKi, a larger number means it's a negative log, so this larger number would have been originally 10 to the minus 8.8, but you turned it into pKi, so it's 8.8. So that means that this has essentially a very low KD, very high affinity, okay? So the bigger the number in this table, using this language, the more affinity there is, okay? So 8.8 and 5.9. Let's compare 2A and 2C. This one's almost minus 9, roughly minus, or this is almost 9, that's roughly 7. So that's a difference of 2. 9 minus 7 is, approximately 9 minus minus 9 minus 6. That's 3, isn't it? I'm sorry. If we called that a 9 and we called that a 6, the difference is 3. So that means that this drug, spiperone, binds 5-HT2A about 1,000 times more tightly than it does 2C, right? Because these are exponents we're talking about, right? So, 10 to the 9 is 1,000 times more than 10 to the 6, okay? So that's quite a difference, isn't it? And it's even less selective, or it's even less affinity for 2B. So what do you think? That one has a fair amount of selectivity, right? The second best binder is almost 1,000 times less affinity. So that's a pretty selective drug. Let's look down here at some other numbers. Look at this one, meancerin, 8.1, 7.38. Which is more selective, spiperone or meancerin? Spiperone, right? Because the numerical differences are much greater. And so meancerin is kind of a crummy drug, isn't it? It doesn't have much preference. You can kind of middling, middle affinities, but there's no selectivity. So selectivity is really different than just simple affinity, isn't it? It's like how much preference does a drug have for one receptor as compared to other receptors. And that's really an important pharmacological concept, because in our organs we have a whole array of very similar receptors that are hooked up in the cell to do different things. So you might want to, if you have a non-selective drug, you might want to be treating anxiety, but you end up having a non-selective drug that causes increased blood pressure somewhere else, And nobody wants to have their anxiety cured by having high blood pressure, right? Drugs that are non-selective have all these adverse side effects because they're activating unintended receptors. And that's why this is such an important concept. So this is all kind of real-world data derived from serotonin receptors. And you can start asking, start developing some ideas about this. So ketanserin, here it is again, we're just kind of looking at the same table. It is selective for 2A, it has some reasonable affinity for 2C. So it binds 2A and 2C, but really doesn't bind 2B very much. So the selectivity is selective for 2A and 2C as compared to 2B, right? And you can even get down to the numbers, and you can say, here we have 9, have 7. It's 100 times stronger in terms of affinity for 2A over 2C, but it's something like 10,000 times stronger than 2B. You get the idea? And so selectivity means nothing unless you have a comparison, okay? And you can, so that's ketanserin. Another one is, we'll look at this, here's an experimental drug, RS102221. For 2A, it has a 6. For 2B, it has also a 6. And for 2C, it's an 8.4. Seems kind of selective for 2C over 2B and 2A, huh? But there is this important thing to consider. You know, we can say this is a selective drug, but what we're really talking about is degrees of difference. So where do you draw the line between what's a selective drug and what's a non-selective drug, right? It's a mathematical difference between one set of affinities versus another set of affinities. And these are always kind of relative. So the meaning actually comes from digging down into the numbers and finding out what the differences are. So here's this, my favorite drug I like to beat up on here, sarin, with kind of really crummy selectivity down here. But you have all these other drugs in here that have differing features, right? So a great question would, let's say we find that HT2B is a receptor that's really important for something that you want to treat. Maybe it's good for, I don't know, I'm just making this up, like appetite or something like that. Which of these drugs, of all these candidates, would be the best, most selective for HT2B as compared to these others? And what you'd have to do is go through one by one or line by line and figure out if there are any drugs in here that look like they'd be particularly good for 2B over the other ones, and which one's the best. Well, that's a crummy one, isn't it? Spiperone is bad for that one, because you'd end up activating 2A. You'd like the number to be bigger in the middle column as compared to the other two. So 5-methyl-OT is perhaps a better choice. You know, and there aren't any great drugs to answer that question except for maybe some of these up here, right? But you can, by doing these affinities, these comparative affinity studies, you can figure out which drugs are best for particular scenarios. Okay. And so I think, yeah, we're actually a little bit early. Maybe I talked too fast, but I hope I kind of explained selectivity kind of clearly and, you know, make the point that it has to be sort of numerical. Do you guys have any questions on selectivity? And, you know, I always like to use examples too, like we have a gigantic tub of M&Ms with 5 colors up here, and it would be a great class experiment to have people come up and grab them, and, you know, who wants to be selective? Me, I would just like grab a handful of M&Ms, but but you get the idea. And so something similar is going on with drugs. But it's really important, and the reasons we want things to be selective is that we don't want to cause side effects. We want the drug to work, and we don't want to cause side effects by activating unintended receptors. I guess the other thing I could say really quickly, I'll just, since we got a little bit of time, you know, what receptors are expressed is very important, but I want to just talk a little bit about anatomical stuff. So here's my terrible drawing of a brain. Here's the cortex up here. You might have certain receptors, you know, that are like present in layers like this, and they're in these cells. And other receptors might actually be in different areas of the brain. And so even though the brain in total expresses both of these receptors, they're expressed in different spatial areas of the brain. So the blue receptor is in regions of the brain that control certain things, and this black receptor is in other regions and controls different things. But the problem is, when you give a drug, where does the drug go? Well, typically, the drug goes everywhere, right? So if you have a drug that is selective for the black receptor, sure, it's going to activate all those receptors, but since it's selective, even though these other receptors are in the vicinity of that drug, they're not going to be activated because of the feature of drug selectivity, right? And so humans are great at coming up with drugs that have selectivity. What we're terrible at is getting drugs to go to particular places in the body. And that's a big problem we still have. And it's an area, say you had a non-selective drug that could bind either one of these, but you could figure out some way to just deliver the drug only to this region of the brain, then that bad selectivity would not be a problem. The difficulty is we don't have any such technologies right now, but they're being developed. But that's a different story. So I want to also just mention anatomically different distributions of receptors as also being a very important concept. And that's a pretty crummy brain that doesn't do much right there, but you get the idea. Okay, so if there are any questions, I'd be happy to address them. If not, a little discussion in terms of continuity of exams. Many of you have already experienced a couple of exams by Dr. Ineskevich. I'm going to, adhere to the syllabus and use the similar number of exams or questions per lecture hour. I'm probably going to put in a couple of extra credit questions as well. So if you guys have ideas, I'll put them in. But any discussion that comes up, I might turn that into an extra credit question or whatever. And if there's no discussion, I'll still probably put them in there. But I encourage discussion. All right, thanks, you guys.